Hi, everyone, and welcome to this third episode of Blue Talks from Ocean Citizen Project. So we're just going to wait another minute for everybody to have time to join. Just a little reminder for those of you who maybe don't know, this is the third episode of a three-part mini-series to set the scenes and understand the blue forest, how they function, their ecological value. And today we are diving into a very important aspect of ocean ecology and ocean restoration, which is connectivity. And it's going to be presented by Federica COSTANTINI, member of CoNISMa. She works at the Università of Bologna, and she's quite the expert on connectivity. So let's just wait another moment. Just so you know, the episodes will be recorded, and it will be uploaded afterwards on our YouTube channel. Actually, the episodes from last week, no, it was not like last week, it was 2 weeks ago. The episode from 2 weeks ago and from last month are also recorded and made available on the YouTube channel. So if anyone wants to rewatch, share, it is still possible. So the structure of today is going to be the presentation from Federica COSTANTINI. Clara and myself, both marine biologists, also working on Ocean Citizen Project. We will be present in the chat if you have any logistical doubts or question. If you have questions for Federica for after the presentation, please try and shoot them in the Q&A box so that we can keep track of everything and make sure your questions are attended. And lastly, after the presentation from Federica, you will be redirected to a quiz that we will go through all together, see what we have learned from these sessions. And then after that, we'll get to the live Q&A, attending whatever doubts that you might have shoot in the Q&A box. I think I've presented everyone and we're ready to go. Yes? Federica, it's all yours.
Okay. Good afternoon, everybody. Could you hear me? Okay. Sorry,
Yes,
I have the people that are in the waiting room, so I try to. Okay. So
we'll take care of that. Don't worry.
Okay. Let me see if you see the presentation.
Yes.
Okay. So I can start, right?
Yes, go for it.
Okay, so good afternoon, everybody. I'm Federica Costantini from the University of Bologna in Italy, and in the Ocean Citizen Project, I have the head of CoNISMa, so the Consorzio Nazionale Interuniversitario per le Scienze del Mare, and this afternoon I will speak about connectivity. So I would like to start this talk telling you a story. In 1992, a shipping container filled with bath toys was lost in the Pacific Ocean. But some of the yellow ducks are still washing ashore today, and some of them have made an incredible journey around the ocean driven by marine currents. We could name this as structural connectivity. In fact, structural connectivity quantifies the physical relationship of landscape elements, including spatial position of habitat, geomorphological features of the seafloor, and hydrodynamic flow features impacting movement, pathway, and boundaries. But marine organisms are not plastic yellow ducks. They actively respond to environmental heterogeneity and structure, shaping their movement and exchange between habitat patches. From microscopic organisms such as bacteria and phytoplanktons to large marine mammals, whole marine life moves through the ocean at different stages of its life cycle. During this movement, organisms interact with other species and with the environment. We can call this marine functional connectivity. Functional connectivity is the outcome of organisms interacting with the landscape. It concerns the patterns and rates of dispersal that arise from the way individuals respond to the structural environment mediated by their behavior traits and dispersal success. This phenomenon occurs through multiple types of connectivity, including larval and seed dispersal, migratory movements of individuals, population, or species, and the transfer of non-living matter, such as nutrients and energy, from one location to another. The diversity of marine organism movement is amazing, and their migratory behaviors have very different influences on the functioning and productivity of the sea and the oceans. Drifters, for example, such as the Pleistonic hydrozoa velella velella, this one, travel by taking advantage of the immense force of ocean currents and even wind, which carry them over very long distances despite their limited swimming abilities. The transported species are indeed an incredible mode of dispersal. For example, the remora, here, uses a suction disc on the top of its head to attach itself to animal much larger than them. Clinging to the tail of the whale, the side of the shark, or the back of the ray, it is passively transported over long distances, switching host according to opportunity and needs. These strategies allow remoras to reach a wide range of environments that are more favorable for feeding or reproduction, places that could never reach on their own. Then we can have the great travelers, such as tuna fish or marine mammals, that can cross entire ocean basins and play a crucial role in connecting different seas and regions of the world. They travel thousands of kilometers, linking distant habitats and facilitating the transfer of nutrients and energy across marine regions. Their movement also aided the dispersal of other species, thereby supporting the maintenance of marine biodiversity on a global scale. Another fascinating category included explorers of the continent, like the hermit crab of the salmons. These marine animals connect land and sea and frequently play a key role in maintaining the balance of the ecosystem they inhabit. By growing in one environment and reproducing in another, they transport essential nutrients between the ocean and the freshwater or coastal environments. Finally, we have the unsuspected travelers. This one. These marine organisms are mainly sessile, but often possess dispersal strategies that are poorly known because they are not easily observable. Among them, we have the marine animal forest, that in the last Blue Talk, Professor Rossi explained to you very well what they are and what are the importance in the oceans. And in this case, during this presentation, we will talk about mainly on coral and sponges as marine animal forest. So animal forming forests such as corals and sponges are sessile organisms that can disperse only through their gametes and larvae. Their dispersal success is so linked to their functional traits, such as their reproductive strategies and larval behavior, as well as demographic factors, including larval production rate, the recruitment rate, and population size. Finally, dispersal is also influenced by their interaction with other species, interaction that can be positive or negative, and by their ecological history. So it's not so easy to understand the dispersal processes of these species. So if we can discuss in general terms for these sessile organisms, like I say coral and sponges, the life cycle of these species started from a source of population where larvae or before gametes are released into the water column and then undergo different pre-settlement processes that can be physical, morphodynamics, and behavioral processes that can reduce the number of the larval pool, and so only a small quantity of larvae then can reach a new substrate and settle. After the settlement, post-settlement processes can further reduce the number of individuals that successfully recruit and grow until reproduction age. So, the structure and the dynamics of the population is linked to the first phase of the life of this species. So it became therefore essential to reduce the disturbances that larvae may experience during the dispersal phase and to strengthen the connection between source and sink population. Studying dispersal strategies in marine animal forests is so challenging, not only because of the limited knowledge of their life history, but also to the difficulty of studying them in the field, especially in the mesophotic environment, so in the deep ocean. So because of this limitation, it's currently impossible to track the dispersal of the larvae directly in the water, so we need to use other methods that I will explain to you very soon. But why is so important to study marine functional connectivity? Because several human-related stressors, both direct and indirect, are fragmenting marine habitats and in extreme case even causing habitat loss. This reduces the opportunity for species to move, to disperse, and to encounter mate for reproduction. Furthermore, stressors such as plastic debris and pollution, coastal urbanization, marine bioinvasion, overexploitation, and climate change all reduce connectivity. Since connectivity is a key factor for the success of natural recovery strategies by means of increasing resilience and the resistance of the species to disturbance, it's nowadays recognized as essential for the delivery and the quality of the ecosystem services. Despite its importance, in a recent paper, we highlighted the research on marine functional connectivity has advanced rapidly, as shown by the growing body of primary literature and the increasing number of reviews covering a wide range of themes and methodologies. Otherwise, through word co-occurrence and network analysis, we reveal clear imbalances. In fact, some geographical areas, such for example the North Atlantic and in particular the Western Atlantic, or certain habitats like the tropical coral reef and taxa, for example, fishes, have received disproportionate attention. In contrast, it's evident that deeper ocean regions, in particular mesophotic ones, and key taxa forming marine animal forests like soft corals and sponges, remain significantly understudied. Sorry. Okay. So, as said before, we cannot track larvae and gametes on the sea for these very important organisms like coral and sponges. So we need to find other approach that can allow us to understand the connectivity pattern and how we can study connectivity. In this video produced within the Sea Unicorn, that is an European coast action project on marine functional connectivity, we presented all the methods used to study connectivity. In fact, scientists use a wide range of methods which fall into four main categories: tagging and tracking, chemical markers, modeling, and genetics. Tracking with electronic tags give the most accurate description of species movement, but can only be used only on large organisms, so not possible to use in coral and sponges, for example. This recording device works just like a GPS and allows the tracking of movement over months and even years. Thanks to chemical markers and biological tissue, we can identify where many marine species have lived and what they ate. To do this, we mainly use hard structure formed at an early stage in some species. So also in this case, chemical markers are not really useful for coral and sponges. We can also use a biophysical model that allows the estimation of present and future connectivity for a wide range of species. And in this case, we can use modeling for coral and sponges, for example, but we need to have very detailed information about their life history traits to allow the modeling to be very precise. So despite all these methods, here in the talk, I will more focus on genetic methods, which relies on analyzing the relationship among individuals. So if we are working at the population level, we can study connectivity using population genetics that allow us to evaluate the distribution and changes of genetic variation within and between population. In fact, population genetics will focus on how evolutionary force like mutation, natural selection, genetic drift, and migration shape genetic diversity over time. One of the approach that we can use with molecular tools aims to identify direct links between parents and offsprings and to measure the distance that separates them. In marine species, this is extremely challenging because a huge number of juveniles and potential parents must be analyzed to detect only a tiny portion of related individuals. So nowadays, recent advantage in high-throughput DNA sequencing allow us to analyze a large number of individuals at a relatively low cost. If we can apply this routinely, this method based on next-generation sequences could provide information not only on connectivity, but also on stock size and population structure. Although genetic methods provide only a partial picture of lifetime movement, they offer valuable complementary information by estimating long-term connectivity, averaged over many generations through the detection of more distant genetic relationships. Why we move from population to community level? So when we try to understand what's happened within a group of individuals belonging to a different species in a specific area and in a specific time frame, we can use other molecular tools that can allow us to study connectivity patterns of more species at the same time. For example, in this case, we can use environmental DNA. Environmental DNA can identify all species present at the given site, providing valuable information about the species range and where they overlap. What is environmental DNA? Environmental DNA consists of DNA released into the environment by organisms during their lifetime. So we can detect this DNA in a cup of seawater, in a bulk of sediment, but even within marine organisms. In fact, some organisms, such for example sponges, can act as natural DNA sampler because of their exceptional capacity to filter seawater. And filtering seawater, they also filtered some DNA that is on the water. Using this natural sampler, we can gain information about changes in community structure along environmental gradients. Also, and this is called beta diversity. So what is mean that if we have along a spatial gradient an increase of beta diversity, it means that there are greater difference between communities, so between community in terms of species richness, suggesting that species are more isolated and therefore less connected. In Ocean Citizen, we use this ability of sponges to capture environmental DNA to obtain information about benthic marine biodiversity and to understand the pattern of genetic structuring of several species simultaneously. So now we can move to try to understand why marine functional connectivity is important in the restoration. That is one of the main key approach that we use in Ocean Citizen. Considering marine functional connectivity in restoration is essential because connectivity underpins the survival, resilience, and long-term recovery of marine ecosystem. Effective restoration requires protecting and managing the entire life cycle of target species, ensuring that all critical stages, breeding, larval dispersal, settlement, recruitment, and adult movement, are supported across the seascape. Using marine functional connectivity, we can maximize gene exchange, ecosystem productivity, resilience, and recovery of the species, of the population, and the communities. And then understanding the pattern of marine functional connectivity can also allow us to avoid bad connectivity. So trying to reduce the effect of non-native species and the spread of disease. There are three aspects that we can consider in restoration that are connected to the study of marine functional connectivity. In fact, understand marine functional connectivity can allow us to locate key habitats, such, for example, breeding site, nursery areas, and feeding grounds, and to evaluate migration corridors between them. We can optimize the dispersal, so we can evaluate the source and sink population, and we can identify connectivity hotspot and migration corridors. Or we can also find some refuge areas, so refuge sites from disease, from biological invasion, or from environmental degradation. And also we can evaluate key escape corridors for reduce the effect of these stressors. So considering marine functional connectivity in restoration ensure long-term success of the restored population. In fact, if restored population are not connected to other healthy population through larval dispersal, gene flow, or movement of adults, they may fail to persist. So marine functional connectivity helps identify source and sink population and ensure restored areas receive sufficient natural replenishment. Understanding marine functional connectivity allows also to enhance genetic diversity and resilience. In fact, connectivity enhances genetic diversity, avoiding creating isolated population with low genetic variability that are more vulnerable to disease, environmental change, and climate stressors. Then we can support ecosystem functioning and species interaction. In fact, improving ecosystem stability and productivity. Then we can increase the efficiency of restoration intervention. In fact, understanding connectivity allows stakeholders and practitioners to choose a restoration site that maximizes ecological impacts. For example, areas that export larvae to degraded regions or that serve as important stepping stone in dispersal network. And then for sure, including marine functional connectivity in restoration plans support adaptive management under climate change, because we can restore an habitat without considering connectivity may lead to areas that looks healthy initially, but that can collapse over time due to lack of recruitment, poor genetic input, or disrupted ecological processes. So including marine functional connectivity in restoration can also anticipate future dispersal pattern and maintain metapopulation stability under warming, acidification, and other climate stressors. So at the end, how we can include really marine functional connectivity in restoration, and to do this, we in the Ocean Citizen project, we are working both at population and community level. In particular, we would like to understand the pattern of connectivity and biodiversity and ecosystem functioning to find the best location and techniques for the application of restoration and to evaluate the source and sink population in the restoration areas. And then we would like to use also marine functional connectivity approach to evaluate the long-term monitoring and to assess the outcome of the restoration plan and to provide information on changes in biodiversity pattern and ecosystem functioning. To answer these two questions, we performed different field expedition in the last years in two islands across the Canary Island, in particular in Tenerife, where the restoration plan will take action, and in an island in front of Tenerife called La Gomera. At the population level, we sample two gorgonians, in particular Leptogorgia viminalis and Leptogorgia alberrima, and black corals in shallow and mesophotic environment. For all of these three species, we collect several individuals in different sites to evaluate the genetic diversity of the population and evaluate the pattern of connectivity of these species. This will allow us to identify the more variable population in terms of the genetic diversity, and to select the individual, so the fragment of the colonies that will be used in the restoration plan. While at the community level, we sample in the same sites several sponges. Here there are some examples. For example, we sample Aplyzina aerophoba, Crambe, Axinella damicornis, both in the shallow and in the mesophotic zones. And in this case, our aims is to estimate the beta diversity as proxy for connectivity using these sponges as a natural sample of environmental DNA. So the idea is to characterize the benthic biodiversity in the area using this natural sampler. This is a very new approach and will be very useful also not only to evaluate and to assess the benthic biodiversity before the restoration activity, but also to monitor the effect of the restoration in the future, because we can use this not destructive method to characterize the benthic biodiversity in the restored areas. So data obtained in Ocean Citizen will be used to create an effective restoration protocol that take marine functional connectivity into account, ensuring that the restored habitat will be ecologically connected, resilient, and capable of supporting long-term recovery. I would like to thank all the people that work with me on Ocean Citizen, but also I would like to thank the two cost action that allowed me to work on marine functional connectivity, so the Sea Unicorn cost action, but also the MAF work cost action. Thank you for your attention.
Thank you, Federica. Thank you so much. It was really interesting. I think I have a special weakness for connectivity. This is one of the aspects of the project that really got my eye and attention. So that being said, give me a moment. I'm going to share with you a little quiz that we have prepared, just so we can go over the concepts that Federica just exposed. I'm going to share the link in the chat so that you can all join. There you go. That's the link. And now if you give me a minute, I'm going to share my screen so we can see. Sorry, this would be it. And oh. Yes. So I can no longer see the chat, but I'm assuming you are connecting to the Mentimeter quiz. So first question, what does ocean connectivity mean? I think Federica made it pretty clear, but let's just make sure we have that. I mean, we need an easy question to go in, right? So ocean connectivity, some of you are still answering the quiz. What does it mean? Does it have something to do with how fast ships can travel across the sea, or how Wi-Fi signals move underwater, Or is it natural links between marine habitats, species, and ocean processes? Or how the tides can affect underwater life? So it is indeed the third option, natural links between marine habitats, species, and ocean processes. To the next. So why is this connectivity important for marine life? Does it help animals migrate, find food, and reproduce? Or is it because it allows energy and nutrients to circulate? Is it both A and B? Or does connectivity have little to no impact on marine species? Just give you a moment to answer that one. Also, Federica, feel free to comment or add any bit of information if you feel like it. I think this one is pretty clear for everyone. Yeah. So connectivity is, as the title of this Blue Talk was indicating, it's basically the ocean's hidden highway or pathways. Without connectivity, animals would not be able to migrate, to find their food, to reproduce, especially marine animal forests. That is one of the main focus. It's the main focus of the Ocean Citizen project. As Federica described, the larvae rely completely on connectivity to be able to reproduce. So it is indeed both A and B. Which of the following can reduce ocean connectivity? So which of those would be a threat to the precious connectivity that marine system relies so much on? Habitat fragmentation and loss, or healthy coral reefs, ocean currents, or marine protected areas? So indeed, habitat fragmentation and loss could reduce ocean connectivity, which is why in restoration projects such as this one, or the cost actions that Federica was mentioning, the Sea Unicorn and the Map World, taking ocean connectivity and especially functional connectivity into account is crucial. It's essential to recover habitat fragmentation if there was any, or to maintain coherence between habitats, this connectivity, and ensure long-term recovery of the ecosystems. So it was A. How do restoration projects improve connectivity? How does that work? Do we restore isolated sites? Do we rebuild the links between habitats and species? Or is it by increasing the number of marine species, or only focusing on surface ecosystem? I think everybody agrees on that one as well, right? So rebuilding links between habitats and species at the population or community level is key to improve connectivity in a marine ecosystem. Great. To the next. Someone already got there. So what does a connected ocean provide for the planet? What do we get from a connected ocean? From healthy links between habitat species? Do we get greater biodiversity, productivity, and resilience from ocean ecosystem? Do we get nicer climate all year round, smoother currents, or all of the above, or well, all of the answers to the left, rather, A, B, and C? So Fede, we have a few that replied all of the above. Do you want to jump in maybe? Yeah, do you want to jump in maybe and comment on that?
But so for the nicer climate, yeah, maybe we can have a nicer climate if we have a healthy ecosystem and ecosystem that works well and function well. So in some way could be a smoother current. I don't really know if increasing connectivity can reduce the currents. So maybe is for sure greater biodiversity, productivity, and resilience of the ecosystem and the ocean in general.
Indeed. So we were going for A, greater biodiversity, greater biodiversity, productivity, and resilience. I'm guessing nicer climate. It would depend what we mean or what you understand by nicer climate. Obviously, a connected ocean is a healthier and more resilient ocean towards the changes that we're facing in this context of climate change. But if we are in a polar region, connectivity in the polar region is not going to change the fact that it's a polar region. It has a polar climate. It's not. It will just be more resilient facing changes that come along climate change, for example, in the long term, right? And currents, from what I was able to learn on that topic, currents are not affected. The intensity or the presence of a current is not affected by the connectivity. Rather, connectivity depends on these currents to be maintained or on the changes in currents over time. Then connectivity can slightly change in different areas, right? So I think that was it. I'm going to stop sharing the screen so I can see you again. Yes. Where are you? All right. Here I am. So thank you so much for participating in the quiz. I think we're going to go to Q&A, see if we have any specific question from the audience. So we do have one question from Alejandro. Is this technology also helping to understand the connection between land and marine ecosystems? Or can we use this tech to justify the connection sea, land, and the species linked in between? So when technology refer to marine connectivity, to functional connectivity.
Yeah, we can also study marine connectivity and the interface between land and sea, because there are some species, as I showed before, that live part of their life in the sea, a part of their life on land. So the movement of matters and energy of these species is also studied by connectivity. And with data to justify connection C, so we can use all these tools from tagging to chemical to molecular tools to evaluate the connectivity pattern and the land-sea interface.
Right. I hope that helps address your question. Anybody else has a question or needs some clarification on any of the concepts that were introduced today? You can shoot in the Q&A box. I think I don't see any other question for now. So maybe, Federica, we have some time.
Okay.
So you want to you had a little video saved for later. I think we can share it with the audience.
So this one, and I can share to you the video that I talked about marine functional connectivity, where we can see all the methods that we can use to study connectivity. So I will open this. You see?
Right. Yes. Yeah, Daniela just typed in the chat. We'll go to your question right after the video, Daniela.
Okay.
Thank you.
Okay.
From the drift of tiny floating plankton to the migrations of large predators, like tuna, sea turtles, and birds across oceans, Marine functional connectivity describes all the movements of all marine organisms throughout their lives. This includes krill moving up and down in the water, the feeding dives of penguins, the long-distance seasonal migrations of whales, and also the lifetime journey that eventually separates offspring from their parents. For many, this separation occurs during the first moments of life, when tiny eggs and larvae are transported by oceanic currents, sometimes over very long distances. So the final length of the journey can't be predicted based on species size or swimming ability. This, coupled with the difficulties in observing marine organisms in the vast ocean, makes inferring the movements of all marine organisms over their lifetimes a massive challenge. To do so, scientists use many methods which fall into four main categories: electronic tagging and tracking, chemical marker analyses, genetic studies, and modeling. Tagging and tracking with electronic tags give the most accurate descriptions of species movement, but can only be used on larger organisms. These recording devices work just like a GPS and allow the tracking of movements over months or even years. Features of the environment like temperature can also be recorded, providing valuable data on swimming depths and the habitats where the animals live and feed. Thanks to chemical markers in biological tissues, we can identify where many marine species have lived and what they have eaten. For this, we mainly use hard structures formed from an early age, like fish ear stones, shells, or vertebrae, which grow in layers, producing rings like those seen in trees. The chemical compositions of these rings reflect the past environmental conditions, which can retrace individual movements at different ages. We can also exploit chemical markers in soft tissues to explore species diets and quantify energy and matter transfers within and between habitats. As DNA is the building block of life, genetic methods apply to all organisms. Recent advances now allow the routine use of genetics to describe the distribution and long-term connectivity of populations and communities. For example, environmental DNA can identify all species present at any site, providing useful data about species ranges and where they overlap. We can also measure current and ancient population connectivity by examining genetic similarity among individuals from different locations. It is even now possible to estimate connectivity across generations by identifying parents and their offspring and comparing their respective distributions. Lastly, dispersal and environmental niche modeling allow estimation of present and future connectivity for a wide range of species. Most modeling approaches copy oceanic currents and environmental conditions and combine these physical data with species physiology and ecology to make predictions of probable species distributions. We can also model plankton dispersal and the journeys of the many eggs, larvae, and tiny organisms it contains. With increasingly powerful computers, these predictions are becoming ever more detailed and reliable. Through combining techniques, we can obtain unique insights into the relationships between marine organisms and the diverse ecosystems they inhabit. This knowledge of marine functional connectivity can then be applied in decision-making for marine conservation and management. For example, in the design of effective networks of marine protected areas in which human activities are carefully regulated, or to better understand where and when fishing controls should be applied to prevent species extinction and fishery collapse. The future promises further integration of the approaches used in marine functional connectivity research and a better understanding of how the movements of marine organisms contribute to the functioning of our planet and regulation of our climate. This will be essential if we are to sustainably exploit marine resources while maintaining the health, biodiversity, and productivity of the ocean.
The initials
Okay.
GPT stand for Generative Pre.
Okay. Okay.
Well, thank you. Thank you. It's interesting to see that there's different branches within marine functional connectivity. So we have a question in the chat from Daniela. She wants to ask about the specific case of artificial reefs. So she's currently studying marine science, and we have discussed their use as a restoration tool. On one hand, they can promote habitat recovery, but on the other hand, if not well planned, they could increase fragmentation and alter connectivity between ecosystems. So from their experience, from your experience, Federica, how do you assess the use of artificial reefs in terms of ecological connectivity, and under what condition do you consider it appropriate to implement them?
Yeah, thank
That's
you,
a great
Daniela, for
question.
this question, because it's really part of the Ocean Citizen to try to find the best way to use artificial structure to restore ecosystem. So what we are trying to do is to evaluate the connectivity pattern before the implementation of the art structure in the sea. So to find really how the natural population or the remaining natural population are connected, and to use this artificial structure are like a stepping stone to increase the connection between natural population, and at the same time to give to the larvae to the gametes of these species to find a new substrate where can settle and where can grow and reproduce. So I think that the use of artificial structure is one of the key methods nowadays to restoration, but I think that if you want to use this correctly, you need to have a very good assessment at the beginning, and including all the parameters, not only the connectivity in terms of pattern of differentiation among species and population, but also in terms of environmental parameters. So we can study connectivity also analyzing the current integrating the current movement and the movement of the organism. We can have a very complete picture of the connectivity in the region, and based on this picture we can define where we can put the artificial structure to implement to foster a new connectivity in the area. I hope that this will answer your question, Daniela, but I'm happy to chat with you also in another moment if you want.
I think
so the
that's
idea of the
question,
Ocean Cities,
yeah.
sorry?
It's,
So the
I was
idea,
gonna say it really reinforces actually what we're doing in Ocean Citizen.
yeah. So the idea is one of the key aspects of Ocean Citizen is to develop a good protocol for coral restoration and other species restoration, starting from the assessment to the monitoring. So another essential part of the restoration is also the monitoring afterward, and sometimes in most European projects there is very few time for monitoring the action, but in Ocean Citizen we trust on the idea that this artificial substrate that we will put in place will be monitoring to evaluate their effect on biodiversity.
Great. Thank you, Daniela, for such a question at the center of why connectivity is important to take into account for your restoration project. So I don't know if you have other questions. Alejandro is asking if he can get in touch with you, Federica. So I don't know if you have a work email that you want to share in the chat, or we can.
Yeah, I can send. I can
whatever
put.
You prefer.
I lost again the things. Okay. Okay, this is my email. Feel free to reach me whenever you want.
That's great. So, well, I don't think we have other questions. If so, please shoot before we close up. But in any case, Federica, thank you so much for joining on the Blue Talks and giving us precious insight on some things such as important as marine connectivity, marine functional connectivity. And well, this was the last episode of the 3-part mini-series, but keep your eyes open and your boxes in your email accounts open as well. Because yeah, I'm just starting this here. We will have another webinar series, also probably a 3-part mini-series in the spring. Okay, so you will get an email to let you know about that. And we hope to see you soon online. If there is any question, you can reach out to also info. I'm going to type it here, info@oceancitizen.eu. We're also available here or through my email address that you have through the following email that you will receive. And yeah, I think that's it. Thank you again, everyone, and see you soon. Ciao.
Bye.